Hello, everybody. Welcome to today's Mindplex Pubcast. These Pubcasts usually promote the latest podcast. We're doing something a little different today because today's podcast is Cory Doctorow talking about choke point capitalism. I encourage you to go watch that, which was also launched today on the Mindplex podcast channel. But today we are bringing Kennedy back. Kennedy Schaal, the Fly Queen, as I get all rock starry when I'm around Kennedy because I've been discussing her like this larger than life super person with Ben since I believe 2007 or 2008. It was one of the first conversations that Ben and I had when we first met. I think it was a Virtual Worlds conference in San Jose or something stupid. Well, it was a stupid conference. There was all this fake stuff everywhere. And Ben was very clear that the technology he was talking about at the time was only half fake. And that made him very different than anybody else at this conference. And one of the things he talked about, one of the real things he was clear that was working now at the time, was this long-lived fly research. How do you say it, Kennedy?
Long-lived.
Long-lived. Then that's the way it is. And these are flies that have been bred to live, I think it's like 5 or 6 times as long now, depending on all these different factors, which is why everyone goes when you try to say, well, how much longer are they actually living? And it's teaching us a lot about our own genetics. And it's really exciting stuff. So we're going to show this clip really quick from the last show, just so you can see how excited I was, and we can pick up where we left off. Rainer, play the clip.
If the medicines end up turning us into flies, as long as we live a really long time, it's okay, right? I mean, I don't mind having wings in the thorax if I can live 10,000 years.
I would not hate.
Maybe we're not going to play the
clip.
Yeah, but I mean, we've seen this depicted. Should we come back to the clip? I think. Okay. Okay. So where does, oh, you mean it's playing right now? Oh, okay. Well, we'll go ahead and show it then if it's okay. Thank you. Pretty truthful. In the future, yeah, you don't have to pause it.
End up turning us into flies, as long as we live
play
a really long time, it's okay. Right?
Come on, just like you did.
I don't mind having wings in the thorax if I can live 10,000 years.
And we're back on.
I would not.
So we're going to come back and we'll be hot on the trail of fly. Yeah, but I mean, we've seen this depicted, I think, pretty truthfully and accurately in Hollywood movies, and then we'll be ready to show that picture probably pretty soon. I would say sort of the pus and the regurgitation and little things like that might take away from the experience of being a fly that never dies.
Fruit flies are much cleaner than that. They don't do that. They're not gross like that.
Oh, excuse me. Those are just household flies.
This is my fly bias coming out. My fruit flies are very, they're pristine. They've got great they've got great little personalities. That one of my favorite things to watch is when I do mating assays, and I will have to watch these male fruit flies do these very intricate dances to try to woo these females, and it's crazy what they go through. I mean, obviously we see a lot of parallels in human life. I mean, we all know like things that we've done for potential partners, and
wait a minute, they have to woo the flies are wooing their partners.
It is a very intricate process.
Yes. And I mean, if you take a middle-aged fly and you feed it,
that's very complex behavior.
If you take the middle-aged fly and you feed it some of the herbs that we developed previously a long time ago from AI analysis of fly genomics, I mean, it will make the middle-aged fly better at doing the wooing dance for seducing the women. It gets the vibes of a young male fly again.
So when the flies are going through midlife crisis, and it sort of gives them more more wooing ability.
Well, no, if we give them our herbs, it makes them their bodies younger again, and then they can do a better mating dance.
Oh, it literally makes their bodies sort of younger again.
That's one of the.
Then they will get laid more than the other middle-aged flies, right? I mean, so there's, you don't want to overplay it. There is, of course, big differences between humans and flies, which is why we need advanced AI to do the transfer learning, all the data from Rejuve network. But there is, in the end, they're eating, they're mating, they're stressing their hearts and their brains and immune systems. I mean, there's a lot of similarity among all the sort of macroscopic organisms on the planet.
They have complex emotions too.
Well, that's what I was reacting to earlier.
Complex emotions and social structure.
Because now we're talking about fruit fly emotions. And I got to tell you, this is the first time I've ever been talking about fruit fly emotions.
Well, let me tell you, as somebody who's watched a lot of fruit flies mate, I can tell you they absolutely are very emotional.
Are they fickle?
Well, that's one of the assays that we do when we're screening therapeutics is you put a female in a vial with two males. One male has received the therapeutic and the other male has not, and you see which one wins for the mating. And
that's not just fickle. That's just, she's doing the best with what she has. You give her two to pick from. She's got to pick one of them. It doesn't really, she may not like either of them really, right?
Yeah, but the treated males are successful beyond what we would expect due to random chance. So you have something that actually makes them
So she knows. She knows which fly is going to, no, wait a minute. Are they having fly sex? I thought flies just. Oh, really?
Of course.
Well, I had no idea that this was going to be an R-rated podcast.
Fly. Oh yes, and fun fact that I learned from my mentor Michael Rose at UC Irvine, the original king of the flies. So when a male and a female mate, the males are much smaller than the females. So the males, once a female like consents, the male just kind of jumps on her back and she sort of goes about and does her business this whole time. They mate for about half an hour, which proportionally to a fly's life is about, I remember Dr. Rose telling all the guys at UC Irvine who thought they were, hot stuff or whatever, that he's like, that'd be the equivalent to you going for 2 weeks. So,
Yeah.
All right. So they got some fly stamina. So I think the memes alone from this episode are going to be quite. I'd like to thank everyone for that.
Yeah. I think I've got to go to another face-to-face meeting here. So on this high note of,
I was going to say, you don't have more time to talk about fly sex. I mean, we're obviously going to have to do a whole pubcast on this now. So as you can see, we were very excited at the end of that video. Ben had to go, so there was nothing we could do. But I wanted to pick up where we left off on that. And basically, it's this really cute idea where, I don't know, it's kind of hard to think about what I thought before, you told me about the fly courtship, because that's all I can think about now. Rainer, can we cut to that picture for a second? It's on. Okay, so this is in my mind what me and Midjourney came up with for Flies in Love. Of course, it looks absolutely nothing like the real thing. Like I hopefully wouldn't need to tell you that. Okay, cut back to us, Rainer. Thanks a lot. And so that the idea was just that flies have complex behavior and complex mating rituals like many mammals. And I never thought of I didn't think that was true. I don't know at what point I thought that the insects just started laying eggs and fertilizing eggs or whatever, but to me it was it took our conversation about anti-aging therapies and genetics and the fly data that can transfer to humans and all that stuff, and moved into this other realm of consciousness, which is something we also like to talk about on the show, but it has to sort of come up Ben's like me. He doesn't like these whole general, we could argue about it forever. Like, what is consciousness? What is this? What is that? Kind of thing. But whenever we have actual examples of sort of like a certain type of consciousness, like in action, that we've studied for a long period of time, it seems like we can actually learn something, from it about consciousness in general, right? Like not just the flies. And there's a lot of stuff coming out now about everything being conscious, even inanimate objects and things like that. But certainly all the insects are conscious. And even Corey talks about this in the podcast that was released today about an Umwelt. He talks about a book called Ways of Being, and I want to say Bridle. I hope I got that right. He was talking about a flea Umwelt. So a flea is looking for blood and then wants to know how much blood. And he walks, that's his life. He's a flea, right? But there's still an Umwelt. There's still this consciousness, this being, this thing that's there, even for something like a flea. So if you got something like a fly, if from my perspective, right, it seems kind of in between, right? Like not quite a flea, but not as sophisticated as us. But it turns out they're still very sophisticated. So tell us a little bit about that before we go back, because I really want to hear your story too about how you got into all this stuff, because it's such an interesting story. But while we're hot talking about sort of like the basics of the fly courtship, I wanted to ask you a few details about that. In fact, I think actually Grace has a question about the fruit fly courtship. Grace?
Yes. How did the results of the AI analysis of Methuselah Fly data inform the development of the supplement?
Okay.
Well, I mean, I can run with that.
That
question.
was the wrong. Yeah, that was okay. I
Sorry.
think, yeah. Go ahead and ask the question on this topic, Grace.
Yes. What specific behaviors do male fruit flies use in their courtship displays?
So, male fruit flies have these really amazing courtship dances that they do for the females. So when they pick a female that they are interested in, they do these really elaborate dances. They'll try to stay within her eyesight, within her field of vision, and they will dance around. They'll flutter their wings. They'll turn circles. They do this whole dance trying to impress her. And sometimes she'll walk towards him a little bit, kind of act like she's interested, and then turn her back and like run away. And he'll often chase after her. No, they're very persistent. They are very persistent. And so they'll often chase after her and then try to get in front of her again, her field of vision, dance, flutter their wings, show off and stuff. And then if she is receptive to the male, she will then allow him to mount her. And so like he basically hops on her back and then she goes about her business, like kind of just crawls around the cage, like eats, like does whatever she wants while the male's like on her back.
He's like holding on.
Inseminating her. Yeah, well, because they mate, like I said, in like the clip showed, they mate for like 20 to 30 minutes on average, which if you do the math and like tease that out to like the percentage of like an average fruit fly lifespan, it equates to about 0.05% of their lifespan, which if you then like carry that over to like an average human 75-year lifespan, that works out to about 2 weeks. So
yes,
it's like, so it's pretty impressive. And like,
yes.
And one of the reasons why they have to mate for so long like that is because the fruit flies actually have really, really long sperm cells, which is really interesting. So the Drosophila melanogaster, which is the type of fruit fly that I work with, which is your most common laboratory fruit fly, their sperm cell is 1.8 millimeters long. Now, the male himself is,
I was going to say, isn't that as long as the fly?
Right. Exactly. And so the female reproductive tract has to be about that long, a little bit longer too. And she's only about 3 millimeters long. And so, there's a tightly coiled system and stuff that takes up all the space. But that's one of the reasons why they have to mate so long, is because their sperm cells.
Oh, because it's long?
They're so large relative.
So it gets the job done, though, is the point. If
right.
And they give it a half hour, but she's going to get fertilized.
There's one species of fruit fly, the Drosophila bifurcata, and their sperm cell is 58 millimeters long, and it's actually the longest, one of the longest cells in the animal kingdom relative to body size.
Wait, how? There's a fly that has it longer than its own body size?
It's a type of fruit fly, so it's a type of Drosophila, and its sperm is 58 millimeters long.
So what? It's wrapped up like an intestine in there or something?
Yeah, basically. Yeah.
Huh.
Yeah.
Interesting. Wow.
Yeah.
Okay.
It's one of the longest cells in the animal kingdom relative to body size, and so it's like absolutely. I mean, to me, it's really interesting. And female fruit flies can actually they store the sperm for later, and then they fertilize their eggs as they come out, so they can store sperm.
Oh, so there goes my theory about needing a half hour to fertilize the egg. So she's going to get fertilized for sure, because she's storing it. It's like she's got the turkey baster built in.
Yeah, but another thing that they can do, though, is a female can store sperm from multiple males, and then they can pick the sperm that they want to use for their eggs.
It's a dream come true, really. That's amazing. And they're gone though afterwards. Like they've done their job, and she's done with them. There's no fruit fly family afterwards when the fruit fly is born, any of that stuff.
No, they're not. Okay. They're not like that.
Okay, I didn't think so. Just checking. Yeah. Okay, great. Hey, Grace, you had another question, I think.
Yes. How does the male fruit fly recognize a potential mate?
All right. Well, that's a great question. So it's kind of fun. There's actually a lot of research that's come out recently on the fly brain and the connectome. I don't know if you saw, like a couple weeks ago, huge study came out that they completely mapped the brain, the connectome of a fruit fly larvae, and it's the most complex organism that has had their brain completely mapped. And in that brain, you can see the connections between the areas of the brain that are related to learning and memory, and how it is similar to higher order mammals. And so fruit flies have a structure in their brain called the mushroom body, which is unique to insects, but the best human approximation of that is kind of like the prefrontal cortex and the hippocampus working together. And so basically the mushroom body does what those two regions of the brain would do, in that they take visual clues, they take pheromones, and they use that information to make decisions. And so a fruit fly can go to a female, and if she smells good to him, and he confirms that she is in fact female, and then he likes her. I mean, there's a certain element of he likes her, because obviously there's the cage is roughly half female, half males, and they're very particular.
There's other fish in the sea.
Right. And so they take the visual clues and the pheromones, and that gets processed in their mushroom body part of their brain, and that is what determines their social behavior and their behavior of going after their particular female of choice. And in turn, the female decides whether or not she wants to mate with that particular male.
Okay, so it's a little bit random in the sense that they're looking at whatever males are available, like you were talking about when you do your testing. They're basically in a test tube,
right,
with two of them. Ben was talking about a specific experiment where you were given the supplement to one of the male flies,
right,
one of the middle-aged male flies, and not the other middle-aged male fly. And so one of them was like doing the dance like way more energetically than the other one, and in fact every time she picked the energetic suitor.
Yes. So I mean, that's a display of fitness, and that's what you just described as one of the assays that we use when we're testing a new new potential therapy in fruit flies is the mating assay. And, you know, it's important because obviously, so if we develop a therapy that somebody might take throughout the course of their life, we want to make sure there aren't life-altering side effects, which I think for most people would agree that would be one of those life-altering side effects that we want to either keep how it is or improve it if possible. And so that's an important assay, and it's part of like the you know, it's part of the way that fruit flies allow us to holistically study how a therapy might affect an organism throughout their entire lifespan, so we can see if it affects their mating at all, and if it doesn't, then, you know, it passes. And in this case, like the particular supplement that we developed actually encouraged more mating success for those males who had been taking it. So
okay, great. Grace, you had another question about the courtship, I think.
Yes. 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 Yes.
Uh-oh.
Yes.
Grace.
Yes. Yes. Yes.
Okay.
Yes.
Well, Grace is having a network moment here for those of you who are old enough to remember that old movie. So we'll let Grace feel better in a minute, and I will go on and ask the questions that I was wondering about. So with the courtship, how long does that usually last? They do it for how long?
The courtship dance I've seen last anywhere from like 5 minutes to about 15 minutes. So yeah, usually after about 15 minutes, a female will decide yay or nay. But
okay,
but if you count, but in that respect, if you like count, like, you know, all the way from like the beginning of the courtship to like the end of fruit flies, it could be like an hour, which is like,
so he just goes on to another one if he like gives it his all.
Not always, and it usually there are studies on this. The males who get rejected do have less mating success over the course of their life, and
because nobody else wants him either for whatever reason.
And there's been some theories because they've studied like virgin males, so males who are trying to mate for the first time, and the ones that are rejected from that first encounter do not, over the course of their lives, do not have as many mating encounters as males who are not rejected in that first encounter.
So they've really got less stamina, or
they're really, I mean, it's a evolutionary biology, it's a selection
in action.
It's a it's a measure of fitness. Like,
do you think that, because two things popped into my head right then. On the one hand, she's maybe detecting what we've already talked about, how there's less stamina, less energy, stuff like that. Do you think there could also be something genetically inferior with these flies that have less stamina, and she senses that somehow in terms of evolutionary biology, picking the best?
I think there's a genomic and, like, you know, pheromone quality connection that the females are selecting for. Yes, because, you know, when you're an animal and you're subject to selection, and I mean it's not just like insects, because I mean humans do this too, right? It's like we all want to have our next generation with somebody who is physically fit, who is like, I mean, physically, but maybe like has better genes, like, and granted that doesn't always go into human selection of a mate, but there is kind of like a baseline of that, you know, people have a type that they like typically, and that and those similar types are usually created by similar genes, right? And so so in a fruit fly, I think there is a genomic connection, and I mean the genes of a fruit fly is going to have an influence over the pheromones that they produce. So
God, interesting. Okay, cool. Well, this is a good time to like I said, because you have a really interesting story in terms of starting out, you went to University of California, Irvine, and you don't, huh?
Go anteaters.
Go anteaters. And you've done this very intense research for many years that it looks like a lot of other researchers are now going back to and duplicating and doing other things, but they're when I was looking around, it looked like they're basically doing what you already did. And going, yep, it did what she said it did, and now we're gonna maybe do this and maybe do that, talking it up, but it was still kind of doing what you already did. So I mean, what's the process going through your mind? You were an undergrad, you hadn't gotten a PhD. You just told me last night for the first time that you actually don't have a PhD, which is incredibly interesting because it plays into this narrative that I've been, decided a couple years ago, actually, when I was teaching at San Francisco State, that, having an academic degree just doesn't matter as much as it used to. I mean, I'm not sure if it ever did, is the question that always comes up, but so I'd really like to talk about how you started off. I mean, that must have been great talking to your parents on the phone, what's your goal in life? Well, I'm working on fruit fly research, like you decided to go.
Yeah. The conversation went about as you would expect.
Okay. And then of course, Ben, I know from personal experience how validating it can be when you have ideas that nobody else can wrap their head around, and then you say it to Ben, and Ben gets it completely, and in fact has several ideas for you to get it further, and he doesn't react in such a way like it's a crazy idea. He's like, oh, I see what you're doing, and blah blah blah, and it's wonderful to be like, oh, well, Ben gets it, and it's very validating. So I can only imagine that was very helpful. 2007, 2008. And what was your freshman year?
2003.
Okay, 2003. So you had already been doing it for, 4 years, right? Before, and then you met Ben and took it from there. So just start at the beginning and let us know, how you got here.
So I have always been, I've been a lifelong science and medicine nerd. That's kind of always been my thing. Even as a really young kid, I always showed a very, a pronounced interest in the human body, in diseases, how all of that works, how the genome influences those aspects of human life. And I remember when I was a kid, my most favorite book that we had were actually my grandfather's old medical books. My grandfather was a physician and surgeon in the 1930s, and then 30s through the 70s. He died in the 70s. But we had all of his old medical books, and I used to just sit for hours and just flip through them and look at all the anatomy and read the articles about human development and just medicine in general. And I just thought it was really interesting. And so, I kind of grew up in everybody else who shows an interest in it. It's oh, well, you'll be a doctor, you'll be a physician. And so,
right.
I kind of always just assumed that was what would happen. And so I got to UCI, and my counselor's telling me if you want to go to medical school, you need to get into an undergraduate research lab, and you need to find a faculty mentor who will be able to get you your letters of rec and do all that stuff. And so the first quarter of my freshman year, I took evolutionary biology and ecology was mandatory for all bio majors. And so, but my professor, Michael Rose, I just thought was a very, very interesting person. And the way that he taught ecology and evolution was unique. He taught it, I guess it was something that I hadn't really thought of, the intricacies of evolution. I knew the basics, the best genes come together and that's the next generation. That's the basic things of evolution. But then I learned more about him and what he studied, and he studied the evolutionary biology basis of aging and the aging process, and how life extension happens, and how those genes are involved in subsequent generations living longer and longer and longer. And he's just a cool guy too. Like if you ever get a chance to meet Michael Rose, like he's very interesting. He's Canadian. He's kind of straight faced, but has like sort of a biting sense of humor. And he loves his rock music. His music is like the second most important thing in his life, and he's very, very interesting. And so I immediately booked time with him during his office hours and went and talked to him more. And I was like, I'd really like to learn more about your lab and possibly join your lab if you have space for me. And he was like, oh yeah, absolutely. And so that's when I learned about the fruit flies and how they were used for studying aging. And immediately in my brain, I saw all of these other implications that, you know, people think on the surface, it's like, oh, you're studying aging, you're studying the aging process, but there's so much involved in the aging process. If you think about how organisms age, the way that their cells stop dividing properly, the way that they stop clearing out cellular garbage properly, the way that you basically accumulate damage and you lose the ability to kind of adapt to your surroundings and to kind of be responsive to selective pressure. That's sort of the aging process in a nutshell, and how that causes diseases that people get as they age. And so I just kind of had this like mental explosion of all the implications that studying the aging process has. And it's like not just about this
aging.
It's not about this vain notion about living forever. It's about
not just about
right, not just about. It's about this, like, you know, about living better, about being healthier,
right.
about, you know. And to me, I was like, well, yeah, this is great, because if you by studying aging, you can solve all these other diseases too that, you know,
that stop people from having even a normal lifespan.
Right. Exactly.
Yeah.
Exactly. And so it was kind of this like aha moment for me. And so I got into the lab, and at the time, he had one grad student that was graduating. And so I, you know, started as like a little minion in the lab, like washing glassware, doing all that stuff, just like, you know, feeding the fruit flies, which, I mean, obviously I still feed the fruit flies. I still wash a lot of my own glassware. But and then when he a couple of years later, well, for a couple of years he didn't have any grad students, and so I kind of took on a lab leadership role, and you know, scheduled people for working on various assays and for taking care of the flies. And because the thing about a fruit fly lab is it runs 24/7. Like, it's not like bacteria or viruses that can be frozen.
They don't sleep overnight or whatever.
No, it's a 24/7 process. And so, you know, I took on the scheduling and all that stuff, and various projects that were going on. I kind of.
So you just stepped up and started running the lab. That's what I'm hearing.
And then Michael got a grad student who came in. Her name's Molly Burke, and she's now a professor at Oregon State, I believe. But she came in as a first-year PhD student and wanted to do this really awesome like cryogenics project. So freezing fruit flies cryogenically, seeing what impact it had on their like aging biomarkers and stuff like that. And so I worked with her on this, and we eventually got to develop a whole strain of fruit flies that were really good at being cryogenically frozen.
Wait, are you saying they come back afterwards? What are you saying?
Yeah, they come back, and then we had larvae and eggs that we froze, and then we're able to thaw, and they hatched and became normal flies and were healthy.
Well, you had adult fruit flies that you froze, and how long were they frozen before you brought them back?
Oh, I think we got up to like about a week. Yeah.
When did that happen?
When did that happen?
Yeah, like a year ago, a couple years ago. When did that happen?
This was in like 2006, 2005.
Oh wow! See, I didn't realize we had anything.
It was such a big deal. Like we ended up on it had like a little spot on like ABC News. Like,
oh, okay, so it was a big deal when it happened. Okay,
yeah,
because that's kind of amazing.
Yeah, and I had people calling me and saying like, "Oh, I saw you on the news," and I was like, "Oh, really?"
Yeah.
Yeah, but eventually it was a very difficult project. And so Molly kind of moved on to another topic of studying heat shock proteins, which are involved in stress resistance and inflammatory responses, and also, longevity connected, of course. But that was a really cool project that we like got to work on. And so that was kind of my first thing, doing like a major research project, and actually being able to help.
Was that your first or second year that
yeah, like
that cryogenic thing happened? Holy cow, what a way to start off. Okay.
So it was really cool. And I just really became passionate about it, and kind of had this light bulb moment where I realized I had the potential to write the things and publish the things that other people would have to memorize, whereas if I went to medical school, I'd have to memorize a bunch of stuff that other people wrote myself. and that kind of really solidified my path into going into research instead of clinical medicine, because I've always been I've been lucky. I have a good memory, but I've always kind of felt like just memorizing mass quantities of data was kind of useless. Like, it's like, so what? Like, what's the connection? What's the big overarching theory of all of this stuff? Like, what next? Like, what's like the creative aspect of that? And so again,
this is always the problem with a lot of academia, right? You learn the stuff and then you want to like apply it somehow, and there's like, oh, we don't have a department for that to do that research or whatever, right? Because it's always like catching up. Yeah. Hey, we had a question I wanted to answer while we had somebody that where a question was actually sort of at the right time to ask, even though it's a little bit not what we're talking about right now. But Paul Berger wants to know, he's bringing up Aubrey de Grey's Methuselah Mouse Prize.
Oh yeah.
And he's saying he personally thought it was preposterous when it came out, given the lifespan of a mouse, and wanted to ask you, wouldn't it have made more sense? Well, I guess there is a Methuselah Fly Prize now, but what do you think about the Methuselah Fly Prize? And did we learn? Do you think, was there any knowledge that you gained from that, or was that always also seems like kind of one of these catch-up things, where it's sort of like in sort of a synthesis of everything to date rather than something new? I don't know. Help me out here, saying something tangible.
I admittedly I don't know much about the Methuselah Fly Prize, actually.
Nor do I, obviously.
I know about Aubrey's Methuselah Mouse Prize, of course.
Right. So he didn't do a fly prize. I thought they did do a fly prize later. I think the point is that they should have done a fly prize.
Yeah, they should have done a fly prize. I you know what? I will have to talk to Aubrey about that. Aubrey is on my advisory board for and I've talked to him several times now. I know he does really tend to like mouse research, which is understandable. Like, people, all this groundbreaking longevity stuff happens, and people want to see something done in kind of a higher order mammal, which makes sense. And so yeah, so I don't think that he thinks that the Methuselah flies are like insignificant in that sense. I think he just he thinks like a mouse prize might be just more eye-catching. But usually you
go in the other direction, right? You'd start off with the smaller animal,
right?
Is it animal? Right. Is it the animals or insects, or are insects an animal?
Oh well, yeah. I mean insects are part of the animal kingdom, but
okay, so they are animals. Okay, so that's why. Yeah.
Right. Right.
All right.
I'll have to ask Aubrey about that. I know. I mean, he's read my my papers on with using the transgenic Alzheimer's flies, and then going straight into humans with the supplement we developed, and he was really impressed with those. And then of course he was like, oh, I'd like to get this and test it in a mouse trial. So yeah, but I mean, I know that he does see the value in the Methuselah flies. So maybe it's just cool. He's had the Methuselah Mouse Prize branding around for too long, and so
Yeah.
More of a marketing. I don't know, but that's a great question, Paul. I will ask Aubrey that next time I talk to him.
Yeah, absolutely. And that's also something I was gonna pester Aubrey anyway, because we've been talking so much about the flies and anti-aging and stuff like that. So let's talk about so again, before we move on, so now we're a couple of years into
right. So yeah,
schooling, and so you're like a junior or a senior, and you've been running the Fly Lab for a couple of years.
And so it's kind of time for me to like make that decision. Like, am I gonna, and Michael Rose had already, suggested that I could stay in his lab and do a PhD there if I wanted, or was I going to try to go into medical school? And
because those are your two choices, right? It's one or the other,
choices,
right? That's how they present it to you.
And
yeah,
this opportunity presented itself. Another professor at UCI, Gregory Benford, who's actually an astrophysicist, and he's a pretty famous science fiction writer. If anybody's a hard science fiction
I
fan,
recognize his name.
He wrote Timescape was his most famous book, which won the Nebula Award in 1984. So he's always been a longevity enthusiast, futurist, all that stuff. He had a partnership with Dr. Christina Riza, whose daughter was actually one of my people who worked with me in the Rose Lab, and then she eventually was one of my first lab techs that I hired too, and then she went on to graduate school. And so Dr. Riza and Dr. Benford had this idea to actually take Dr. Rose's research with the flies and spin it into a real biotech company and try to actually create anti-aging therapies and treatments. And they wanted somebody to come and run their lab and design experiments and write their papers and do all that stuff. And Dr. Rose recommended me for the opportunity. And that meant that since I was going and working for another company, I actually could not get a PhD because it's illegal for you to do that. Because when you're getting a PhD in hard science, you are an employee of the university and they like own you. And so I kind of had to choose between those two things. And I had a long conversation with Dr. Rose, and he's like, well, do you want to teach ever? And I was like, really? Like I see myself like as more of like a guest lecturer, like here and there, maybe sometimes, like, but no, I don't really have an interest in teaching. And he's like, well, then you really don't need a PhD. And so I was like, okay. And so I went and I became the lab director and senior biologist for what became Genesiant. We started right away. Our first project was sequencing our fruit flies, so the control flies, and sequencing the Methuselah flies that we had at that time. And that's where Ben came in, about 2007, 2008. So we had those genomes sequenced, and Ben was able to combine that sequencing data with data from the Welderly database and other human genomic databases that were available at the time, and found not only genes that separated the Methuselah flies from the control flies, but also found human orthologs to those genes. So an ortholog is a gene where the sequence and the function are more or less preserved between two species.
Oh, okay.
Right. Yeah. And so
that's what we're looking for with this research.
Right. Yeah. That's what we were looking for.
Being able to transfer the knowledge, like you were talking about.
Ben's original report on this said that the Methuselah flies display an embarrassment of riches. Was his exact wording in the report, an embarrassment of riches for studying human longevity.
Embarrassing in that we hadn't found it till then.
Right, and that there were so many options, like, basically. And so we actually had to pick what suite of genes we wanted to stay. So there were genes like involved in neurology, cardiology, metabolism, all of that stuff. And so obviously Alzheimer's and related dementias are something that has been a problem for a long time and is only becoming more and more of a problem as we have a larger aging population. I myself actually lost my own mother about 3 years ago to Lewy body dementia. And it is something that I wish on no one. Like, it was a long 10-year degradation process, and it was just so hard to see my mom, who was this like gorgeous, brilliant, vibrant person, kind of not just, it was really, really sad. And so and that happened, like, I decided to study aging and study all this stuff before my mom's diagnosis and before all that stuff happened. So it was kind of a coincidence that happened, but it's
very common though,
right? And it's definitely further like galvanized my passion for the study of aging related disease, for sure. So we decided to go down this path of neurologically related disease. And Ben.
So you've met Ben at this point.
Yeah, I met Ben at this point. So
how did you meet Ben? How did you meet Ben? How did that happen? Did he reach out after reading the paper, or?
Oh, no, he came. Well, we wanted him to do this analysis, and I think he knew Gregory Benford, I think, is who he knew initially. Just because Greg has run in futurist circles for forever. And so Ben came over to Dr. Riza's house in Newport Beach in Orange County, and that's where I met him for the first time. Yeah. And I immediately thought, obviously, Ben's an interesting guy. You can't like not think that he's interesting.
You can't unsee Ben. It's
you
true.
Can't unsee Ben. Yes, that's true.
It changes everything, and it's a combination of new ideas you've never heard of before that make complete sense. And like I said before, often the validation of ideas that you've had that everybody else seemed to think were too crazy or whatever, but he fits it. We'll fit it all together.
It is crazy when you have that moment that you realize you're literally talking to one of the smartest humans on the planet, and they
don't let him hear you say that. No, go on.
But I mean, it's not a subjective thing. Like, it's objectively. Like,
no, it wasn't. And that's like Amara Angelica and I. It was so funny when she worked for Ray, and she's the one that brought me into this project. And she basically, me and her had both decided in, I don't know, 2010 or 11 or something, that if we ever got a chance to work with Ben, that's what we wanted to do. He didn't have a project or anything at the time for us to work on, but we were just like, attach yourself to that guy. He's going places, and he's also happy to bring anybody along for the ride. That's the fun part, is he wants to explain it to everybody. And
that's the thing. It's like, I mean, he's so brilliant, but he's also like so talented at like putting people at ease and making them feel validated, and that what they have to say is interesting. And I mean,
because we got to bring everybody in on this whole benevolent singularity thing. And
exactly. So it's just like, for a young recent college grad who is say what it is, it's a female trying to break into a male dominated world and stuff. And I've always had a little bit of imposter syndrome and whatever, like, do I really belong where I am I really like good at this stuff? To have that validation was very, very encouraging and very very nice in my early career. And so so I've enjoyed working with him like ever since then, pretty much. And so he did our first set of target identification, and crossed it with some databases on traditional Chinese medicine and herbs, and found some that should work on these targets. At the time, AI wasn't as advanced as it is now, and so we couldn't do all of that on a computer. We had to actually go and do some actual literature research, and Dr. Christina Riza poured over all these genes in, on pen and paper with pen, and looking at all the pathways and picking the ones that were most likely to work. And so then we did dozens of fly assays, just like literally testing every combination of these substances that we could think of on our flies to make sure it didn't damage their lifespan, to see if it extended their lifespan, didn't hamper mating, didn't hamper egg laying, all of that. We ultimately came up with this like winning combination that we then tested in a transgenic Alzheimer's model. So flies that you could induce Alzheimer's symptoms in, and this combination of substances alleviated their symptoms. And since they were all GRAS substances, we were then able to actually take that supplement and put it in a clinic of Alzheimer's patients with Dr. William Schenkel at Hoag Hospital in Newport Beach. And over the course of 15 months, we had mild to moderate Alzheimer's patients that preserved their cognition and without bad side effects. Like we didn't have any brain bleeding or anything like that you see from some of these amyloid plaque removing antibody treatments that are out there now. But
when you say that their cognitive ability, I forget the words that you used about the cognitive ability,
they retain their cognitive ability.
So they're just not deteriorating anymore.
No. They stayed pretty close to their baseline throughout the course of about 15 months was how long we had the pilot last for. And a lot of those people chose to continue taking the supplement after the pilot was done.
And is that the Rebuilder supplement
yeah,
we're talking
about?
So
it has two names because of various licensing agreement changes. So it was originally MX100A, and then it was sold as MEMX100. And then another company called Genair came and licensed it and sold it as Rebuilder. So yeah, so it's currently Rebuilder now. And then we also we tested it in Parkinson's flies as well and saw the same effects. And then we never did a formal Parkinson's pilot, but we have had people anecdotally use it for Parkinson's with good effects. So
yeah, and it's one of these funny things where it's hard to know what it's doing because everybody kind of degenerates at a different rate. And
right.
I know I have some family members with Parkinson's that are on Cinemet for Parkinson's, and when the symptoms get controlled, they want to say, I'm cured. Everything's right. But you're basically just trying to stop it from getting worse. That's what these supplements are doing, right?
Right. Yeah, that's what, yeah. I mean, that's kind of the best we have right now. And unfortunately, like I'm hoping like now that I'm with SingularityNET, we can, raise more capital and eventually do biomarker studies and see how this supplement actually works, and maybe do more human trials and actually like make a drug out of it instead of like a supplement that actually has like more effective. But it was also the cool thing about the supplement too is we also created it with the idea that people could take it prophylactically. So like I've taken whatever version of it has existed since 2012 or whatever.
I've been taking it since last Monday.
And so
and I thought, what the hell? So again, you're not going to notice.
My own dad, my dad's 78 now, and he's taken it since 2012 too. And he's still cognitively and physically very, very healthy.
Right. So it's one of these things where you may never know exactly what it stopped, what bad things it stopped from happening, but it's better to just take the supplement and you don't have to know necessarily, but we know that it's doing something.
That story, it's a success. It's good. I am determined to make something really good out of it, but it also, the thing about it is that it really shows the value of the pipeline that we've created, taking these genomic information, using the AI and the machine learning to identify these potential orthologs between species and targets for treating aging, and even identify like substances that could possibly act in those targets. Like it was a validation of the pipeline, and obviously the AI and the machine learning tools have only gotten better in the last 10 11 years or whatever. So I think this is really kind of the way that drug discovery is going, and we hopefully will be able to produce more beneficial medicines that are safer and faster.
Right. And this is the point of Rejuve AI and Rejuve Bio working together to throw the machine learning basically at the genetic data that's coming in to suggest new therapies.
Right.
So Grace, you had a question, I believe, about the rebuilder study. And that supplement.
What are the 8 components of the botanical mixture in Genier Rebuilder?
Oh my gosh, the 8 off the top of my head.
Whatever, what the important ones are. No side effects is good. And
right.
So it'll either help or do nothing. That's
so
good supplement.
The main component came from astragalus root, which is has been used in traditional Chinese medicine for forever as a memory supplement. If you get down to the actual pathways it works on, it's a really potent anti-inflammatory. It really helps the body's cytokine response, so the body's response to inflammation. So it kind of preserves how everything works, rather than, you know, when your body, like when your immune system goes haywire and you release all these cytokines and inflammation, like stuff it doesn't work very well. And so it kind of keeps the body's inflammation response like nice and measured, because you don't want no inflammation, because then your body doesn't heal itself. You want a proper amount of inflammation. And so astragalus root is very good for that. And so that's kind of our really like heavy hitter that's in the supplement. There's also a couple of resveratrol analogs in there. A lot of people know the name resveratrol, like, you know, red wine got really popular because it's got
right. It's good for me. Glug glug glug. Yeah.
Spoiler alert: if you drink a bottle of red wine a day, it's not great.
It's a glass a day? I thought it was a bottle a day. Sorry.
But yeah, so it's got a pterostilbene, which is a resveratrol analog that's found in blueberries, and people know that blueberries are obviously very high in antioxidants, very anti-inflammatory. Let's see what else is in there off the top of my head. I mean,
it's okay. It's not a quiz.
I mean,
it's just a question of what's in there. So it's
so
astragalus and other Chinese things that are found in traditional Chinese medicine.
Yeah, it mostly works on inflammatory pathways, is mostly what it works on. Also, one of the targets is AMPK, which is very important for energy metabolism. That's a pretty big pathway that people in the longevity space have targeted with supplements for a really long time.
Okay. All right, cool. Grace, you got another question, I think, about Rebuilder?
Does the effect of Rebuilder vary depending on age, gender, or severity of Alzheimer's disease?
Yes. So in people that were more severe, we did not see as much of a benefit. They did decline slower than we expected them to, but they still did decline. It was mostly the people who were mostly mild, and the people who were kind of like early moderate stage really did the best. Females and males, I did separate that data out, and we saw about the same effects for females and males. One thing that people ask about is ApoE status, like if people knew their ApoE4 status. So ApoE is a gene that that's often implicated in heart disease and dementias, and specifically ApoE4 is what's associated with Alzheimer's and dementia. And usually if you've got one copy of ApoE4, I think it's like you're 30% more likely to get Alzheimer's. If you got 2 copies, you're like 50 to 80% more likely than somebody who doesn't have that gene to get Alzheimer's. And so we did have people who knew their ApoE4 status, and people who were heterozygous and homozygous for ApoE4 still saw a positive effect from taking the supplement over 15 months. The N value was pretty small. I think we only had like 10 of them, I want to say, but they did.
N value is just the people that had no effect.
Number of subjects, and just stands for numbers.
Okay.
Yeah. And so I think we had about 10 of them who knew their ApoE4 status and were positive for ApoE4. And they saw a good preservation of cognition over the course of the 15
So you weren't testing for that status when you got the
data.
You were using people that already knew,
right? So yeah.
Then you're already a step ahead, having the group that you want.
Yeah, we didn't have the budget to do all of that genomic testing and stuff, but as part of their treatment at the Schaal clinic at Hoag Hospital, some of them had their ApoE status was known, so we used that information.
Okay. Yeah, I remember Ben had a biomarker for Parkinson's way back. That it turns out nobody wants to know if they have Parkinson's in their 20s.
Yeah, see, that's the thing. And I always and I get asked that question a lot by like my friends, like, oh, should I go get screened for all these genes? Should I do? And the thing is,
23andMe doesn't even tell people half the things they used to for that reason.
Right. I mean, yeah. And so the thing is that like you kind of need to get right in your head about what that information actually means, right? And so some people, they get back that they're that they have the BRCA gene, or they're like, oh, well, I am going to get breast cancer. It's like, granted, I'm not a cancer specialist, so I can't speak to that too much, but
it's a pretty good chance, I think, if you have the BRCA gene, right?
Right. I mean, it's a good chance, but it's like, it's not like 100% that you're going to get it. It's something to be aware of. Just it's information. So maybe you get extra mammograms, maybe you get some extra things. You just keep an eye on things,
proactively get breast removal.
Yeah. And so right, exactly. So it's information. And then the Alzheimer's genes or Parkinson's genes and stuff like that, if you're aware that you have those, like in the case of Alzheimer's particularly, we know that social interaction, maintaining good physical health, eating well, like all that stuff that everybody tells you to do all the time anyways, actually does really help. and it seems that the maintaining a good social life really helps more than anything else in terms of keeping your brain healthy and functioning. And so, you see that you maybe have these genes, you maybe have like more propensity to have these things. Okay, so you start walking every day. If you don't, walk with a friend, walk with several friends, make sure you maintain those relationships throughout your life. Meet people to play cards or pickleball or whatever, take cooking classes, like keep learning things, like all that stuff really does help. Like it's people think it sounds
overall
kind of everybody wants this like magic bullet, but until we're looking for that, until we get that magic bullet, these are things that really do help. And having worked with a lot of Alzheimer's patients throughout the last 15 years or so, I can tell you like the difference in the people who really try to maintain those aspects of life and the people who kind of just like sit on their couch at home, their disease trajectories look completely different.
It helps. It does make a difference. Grace, you had a question along these lines.
What other medical regimens can be paired with Rebuilder to optimize its effects?
Well, thanks, Grace.
But medical things, not just, having a good,
medical things.
Psychological health from meeting your friends and stuff like that. What other stuff?
Keeping up with your doctor, making sure you have a good neurologist that you're seeing and stuff. And they will prescribe the standard of care now is usually there's Namenda, there's rivastigmine for various forms of Alzheimer's. So we had everybody who was in our pilot stayed on their regimen that they were on. We made sure their regimen didn't change during the 15 months. So the time that they started taking Rebuilder, like everything stayed the same. And then it was just added on top of their normal medications. And then, if you want to monitor you can monitor your MRIs, like what your brain looks like. You can see what the dark matter looks like. There's tests.
But what do you do with that information? You've got your MRI. Here it is.
Well, you can see. It helps you see like the status of your brain. So is your brain like sometimes there's brain shrinkage that happens over the course of the disease, and so you can have your brain.
Okay.
You can monitor your brain volume.
So to see if there's improvement.
Right. So
and
then when you keep up with your doctor will perform cognitive tests, the functional cognitive tests, and so that will give you an idea of how you are performing cognitively. And they're like memory tests and like remembering the names for things and seeing like a bunch of animals and remembering what the animals are and like things like
like the Trump IQ test. Cow, snake, giraffe, right?
Yes.
So I just want to remind everybody that we are taking questions live. Ask them in the YouTube chat if you have questions. And also please remember to subscribe to the Mindplex podcast, as well as the SingularityNET channel, which we're broadcasting on right now. Mindplex podcast channel on YouTube. So let's talk a little bit about how the machine learning, basically the open cog machine learning algorithms that are combining sort of with the fly genome data and that whole thing, and what's happening there.
It's Ben likes to say cross organism genomic analysis in the bio atom space. Yeah. And
cross organism from fly to human.
Cross organism. Well, yeah. Right. And then there's other model organism data that's going to go in there that I don't work on particularly, but that is available. And so what's really great is that we have this, we'll have this wealth of human data coming in through our Rejuve network users. I know Rejuve AI is partnered with some genomic sequencing companies that will be able to take participants' DNA and analyze that. And of course, they'll get Rejuve tokens in exchange for that. And then I will get that data, and it will be analyzed as part of the bioatom space, and it will help us find more genomic commonalities between the flies and humans and other model organisms. And basically, more data helps us find better targets faster. And one of my big hopes too with the Rejuve network participants is that we'll get like much more diverse human data than is usually available in a human trial. I mean, a lot of people know that human trials tend to skew white, and that's a problem that we need to solve. And Rejuve is trying to make that better with, opening up this network to anybody who wants to use it and compensating them for their data and allowing them to have ownership over their data for their however long they choose to participate. And that will really help encourage a real diversity of participants and of data that we will get for our bioatom space to identify longevity and disease targets.
Perfect transition. Let's talk about this bioatom space. We barely started talking about it and actually we did it in episode 3, and then we did it again in the Pubcast, but it's always, while we're kind of in the heat of something else. So I didn't want to interrupt and go, whoa, back up. What's the bioatom space? But Ben's been talking about it a lot lately. And I just want to get started starting for a beginner, and then go ahead and get complicated in terms of what
I'm not gonna get that complicated because I don't understand.
You don't have to get that complicated, but yeah, the point is we're trying to include everybody following along, because you always have to start somewhere when you're starting out, right? And then you can take it from there. So just give us a kind of bioatom space 101 kind of thing.
Our bioatom space is like, it's a as I understand it, it's comprised of these deep neural networks. So the structure of AI that SingularityNET likes to use, where something isn't just like regurgitating data, but actually learns and reasons from the data that it has. And so the bioatom space, could really help us identify these genomic targets and substances and drugs, and potentially small molecules and gene therapies that could act on these pathways. And it's something cool that happens something cool, like it's mind blowing actually, especially like with the recent advances in AI, that we're talking about, like we have the potential that we could basically discover an entire therapeutic using all of this data in this neural network. And it's crazy, rather than just going through like, the fly trials and the mouse trials and the monkey trials and like, oh, does this work? And then doing all these like little like analyses and stuff. you have this like wealth of data that gets
You're saying the AI is able to put together connections between the data that you're feeding in that normally would require additional trials.
Right, exactly.
For our little brains to come up with it.
Right.
Okay.
Right. But
okay.
This can just kind of
and you know it's right, because of course the point of the trial is that you're sure, so to speak, because it's actually happened in X number of patients. When
right. Well, that's where the data all comes from that it's trained on anyways. So
but the data that it's taking it are from patients that weren't necessarily given the exact same
but
thing.
There's all but it also is trained on the data that is existing from previous trials on previous substances and stuff like that. So
okay, so again, the data is there. This was actually another thing that that Ben and I used to talk about when we first met. The National Institute of Health has everybody uploading their records, all their test data, but there's no standardized format for it. So you get to try to make it work, but that was another idea that Ben wanted to point an AI at all the existing data, right? Like information that's already there, but we just didn't know how to look at it right when the data came in the 90s or whatever. Right. So the bioatom spaces are taking into account previous research, current research, and then is it actually suggesting new trials and things like that?
I think that's the goal. Yeah, that's the goal of the bioatom space. Yeah, to kind of guide to eventually be able to guide our research, and
so it's making suggestions every step of the way, basically for everything. Okay.
Right.
Neat. And so let's see. Trying to see now what we still have to talk about. So when you are feeding information, I guess I'm still trying to figure out this pipeline of how the information that's coming in all the time. So I've got my Rejuve app, and I'm actually didn't join the beta in time, so I don't have it, but I'm being hypothetical, so I won't say anything stupid about the app. But the idea is that I'm getting tokens. I'm building up tokens that I can use in future therapies. It's taking my data into account. So that's another feed in the pipeline is what I'm trying to say. But that's going in. So there's a number of different feeds that are all going into these bioatom spaces, and it's just crunching away, making new recommendations on based on whatever it has at the time.
And it's a generative model. Like it's like Chat GPT, like how Chat GPT is built on, but it's but for bio data, and so it's like a fine-tuned model with genetics, and so it'll take like all that information and generate more information and insights out of that. So if you're using the app and you're putting in your health and wellness data, and then eventually like your genomic data or whatever, and getting those tokens, and all of that data goes into this generative model that helps us find insights for further creation of therapies and supplements and other things that will help us live long and healthy.
Cool. All right, neat. Grace, I think you had another question about it was the Alzheimer's study where they were still calling the supplement MX100A. What was that question you had?
Yes. Has MX100A been tested in any other animal models?
Not that I know of. No, I don't think so. Just the flies, and then I know it's been tested in stem cell models in like petri dishes. So in vitro, it's had quite a bit of in vitro testing done.
Okay. All right. I think you had
one of my former colleagues, Bryant Villampanto, who worked with us on developing it, is a very skilled microbiologist, and so he did a lot of the in vitro studies.
So
informed fly
this
studies.
just went straight from a fly study to a human supplement. There weren't any tests on humans.
Not before, yeah, because I mean, that's one of the advantages of making something that's completely comprised of what the FDA calls
TCM.
Grass substances, yeah, generally regarded as safe.
Oh, they call it grass substances, all the traditional Chinese medicine stuff, that the thousands of years of medical knowledge that's been reduced to grass substances. Like FDA.
Yeah, generally regarded as safe.
As long as they don't stop us from taking it, I'll be happy. That as long as we can, yeah.
So
okay, great.
Yeah, that was the idea, was to use grass substances on our first try, so we could get something into humans as fast as possible.
And that's the beauty with traditional Chinese medicine. I'm obviously a believer, standing up on my soapbox for a moment, because it either helps or does nothing. Usually, there's none of these bad reactions from something that doesn't agree with you. If it doesn't agree with you, it may not work, but there isn't this commitment, this horrible side effects. I think we had one more question about Rebuilder, Grace.
What other studies have been conducted on the effects of Rebuilder on Alzheimer's disease?
So the other studies that have conducted, we pretty much just done this one human pilot, and then people who have taken it anecdotally and reported back to us what they've seen. But the human pilot that we did at Hoag Hospital had about 50 people in it, and it was 15 months long. And I went in there and I interviewed all of these patients and their caregivers every 3 months and gave them a battery of cognitive tests, and plotted all that information and analyzed it. And
cool. All right, we've got some questions now that are coming in.
All right,
so I'll just ask, see what's going on here. So Jake Gonzalez is asking, how can Rejuve fit into drug discovery and development to produce drug supplements free from patents? So this is an interesting question. We talked a little bit about this in the last Pubcast. The supplements are generally free from patents, except when a company decides to try to patent one of them, which happens, every so often. Again, it's a very different process,
because you can't patent naturally occurring substances, I don't believe. So,
well, supposedly you can't, but certain companies have tried. They try to patent their process for synthesizing it or something else to get into that realm. But I think their question was just, I think that they're asking about drugs that Rejuve might create, and those probably would be patented for defensive reasons, even in this environment that we're in. More than anything else, I actually have a friend of mine that companies have tried to sue him over his own technology when they rip it off, and then 5 years later they go back and try to sue him. So he does defensive patents for everything now, which is just it's a record with the government saying, hey, I'm gonna file this thing, and you don't have to even file it. You can just do a provisional patent, and then it's there, and at least it shows prior art.
Right.
But do you happen to know what the plan is with Rejuve in terms of,
patenting
therapies?
I would assume that we would patent therapies, but of course, if you have the patents, you can choose to either silo that in some ivory tower, or you can still make it open source, for lack of a better term, so that people can use it. If you own it, you get to decide what happens to it. So I could see a scenario where we do patent therapies, and then obviously revenue that's created, our first order of business is to compensate the people whose data contributed to that data, which is to that therapy, which is another Rejuve AI network, is so that people who have participated in the biomedical research actually do get fairly compensated for the success that product achieves. but yeah, but I mean, you can always license. I mean, in my opinion, like if we developed something that was like hugely helpful to the masses and was just like groundbreaking in terms of like alleviating suffering and helping whatever aging related disease is targeted, like I wouldn't want to keep that from people, and I wouldn't want to like charge.
Well, this goes into this whole have not thing about these
Yeah.
technologies in
Right.
general, right? Yeah.
Right. Yeah. So I mean, you know, I'm not a patent attorney, so I don't know exactly like how that all would work, but I you know, I wouldn't be like, you know, one of these companies with these like rheumatoid arthritis drugs are usually a good example, because those are the highest grossing drugs that are out there right now, are RA drugs, you know, but they come out with like, these big companies come out with like huge, like really, really tiny little changes to the formulations every like 10 years or whenever their patent expires. So then they can get a new patent and continue to like charge like an insane amount of money for these drugs. And it's just like, I don't have any intention of doing anything like that.
Yeah. You gotta share with everybody.
Like that would like completely like fly in the face of everything that like, you know, SingularityNET and Rejuve and everybody that totally is not in alignment with our ethos as an
our ideals, right? That the idea is that if we have some sort of life-saving drug, that we would share it, and that this is why this is one of the main differences between a benevolent singularity that's taking the planet and people's welfare into account, and just a singularity at all costs kind of thing, where none of that stuff is taken into account. Someone's asking, again, Paul Berger is asking if we use large language models to guide our search on candidate compounds for rejuvenation, whether it's Chinese medicine or, let's see, what is it? This would apply for traditional Chinese medicine too, provided you assembled a large specialized data set. Okay, so this is two questions, as I was warned. So the first question is simple. I'm sure there's some large language models in there just for having conversations, which is all it can do. There's all this freak out lately over LLMs because they are so well at cooking down data and talking about it.
Right. Yeah, I had one friend say to me the other day, she was like, because she was asking about all this stuff and I was talking about it with her, and she was like, so to me, like ChatGPT is like what I have always wished Google would have done for me from the beginning.
Right.
Like
in terms of giving you the answer.
Just give me the answers. Don't tell me like where to go for it. Just give me the answers.
Right. It doesn't matter if it's right. Just give me an answer.
I mean, yeah, kind of like a good like distillation of like, you know, these
and that's, you know, and like for things that don't matter that they can't get wrong, like when you're just writing a website about a band and you're feeding in the information and just when you're just saying the same thing 50 different times, it's great for generating the same thing 50 different ways, you know. As long as you don't ask, you know, because it's had a pretty bad week actually recently at saying researchers research things they don't research, or that somebody's a critic of somebody when they weren't. And finally, drum roll please, it's been accusing some people of sexual harassment behavior on campus and stuff like that they did not do. So it gets a little confused about reality when it's trying to do too much, but it's very good at this kind of thing. And can you talk a little bit about, so first I want to ask the other half of this question, sorry. So they were talking about how the large data set for Chinese medicine would be very large. Okay. So that just, you know, talk about how that worked in terms of like using the data and cross-checking it against, I guess.
Yeah. So I mean, I guess like the
Chinese medicine.
I mean, and again, you know, I am not an AI expert by any means, but yeah, I think at the first, when we did our first passes with the data that was available, and the AI and machine learning tools that Ben used, I think he'd probably call those like kind of a nascent large language model, or a large language model at the time. That But of course now, I mean, what we want to move towards with the bioatom space is something much more fine-tuned, and we have the computing power now, and the good generative models that could handle a data set that large. And like I said earlier, when we found our first targets and our first TCM substances that we used, we did still use a lot of pen and paper research. So a lot of literally just Google Scholar and PubMed and the NIH books, like looking for articles and looking for people who had tested it, and literally going through with a highlighter and seeing
the existing research that had already happened.
Right.
Yeah.
So there was still we were doing like a pen and paper large language model to a certain degree.
Yeah, it's funny because you said pen and paper, but then you talked about Google Scholar, and the Internet Archive has a great Scholar, like thousands and thousands of open source papers now, because we have the loophole I always like to talk about. Graduate students, if you want to publish your paper, just publish a version that isn't quite the final version on your website, and then go put it on Internet Archive Scholar. And that's how we get around the 6 months to a year or whatever that you may have to wait for your final paper to publish.
Right.
So this idea that one of our viewers actually brought up, that was another thing that Ben and I talked about in our that first day when I was just following him around, and trying to look for real world applications of everything we were talking about, right? And one of the ideas is this idea that he had for basically a chat bot. I mean, these are all overzealous chat bots that would have medical data in it to assist a doctor as he's having a visit with a patient, suggesting things and stuff. And as we know, this is a perfect job for something like ChatGPT. It's been getting credit. I talked about the bad things that it did last week, but it also did some good things a week before last, I think. They're saying it saved a dog's life now. It was able to cook through all the data that had been given, and the person fed in the actual medical data from the tests that the vet had given it. And they were able to rule out the things the vet had already tried to do. And they gave it a supplement, I think, even, or it could have been a medicine, but it was something very simple for the thing that it was, and the dog got better. And again, this isn't brilliant ChatGPT doing anything brilliant. This is just being able to sift through all the existing research on a thing and then cross-check that against input that it's given.
They can just do it much faster than we can.
And find relationships, like with that Parkinson's marker. It was looking at subgenes. So we were never, because there's thousands and thousands of subgenes. We can't match them up, but the AI can, and that's what we're doing with all this.
Yeah.
So
I mean, and that's the thing with the fly research too. I mean, a human genome is 3 billion base pairs, and then a fly genome, just a little fly genome, is 160 million, which, I mean, that's still, that's a lot of info. So yes, we need these large language models, and we need these generative AI models to help us sift through all this data and find the important patterns and connections.
Cool. All right, great. Well, thank you very much for coming on, Kennedy. This has been really exciting. Oh, we've got actual flies. Yay. So that's what they look like.
Yep. This is
how
they look.
That's how many are in there?
About 2,000. About 2,000.
Wow. Okay. Yeah. And so you put them in a container and they get a number or something, and
they're bred in vials first. So I let them, the eggs they lay eggs in vials, and then the eggs develop, or I take the adults out of the vials, the eggs developed, and that's like their nursery, these little comfortable little vials. And then when they hatch, then I transfer them to the bigger,
into the larger container.
Yeah.
And then you're just looking for new pairs to breed once you get the new container?
Oh no!
What do you do with the flies in the container?
These are Methuselah flies. These are super Methuselah flies,
right?
And, so these flies are about 4 months old now, so they're just a little over halfway through their lifespan. for the Methuselah flies. So only the flies that lay their eggs at, say, 6 months of age, those eggs will get used for the next generation.
Oh, okay. So
we know that those eggs, yeah. So we know those
in the container. We want to see how long they're going to live. That's it.
Right. And so, yeah. So I take the eggs from the flies that live a really long time, and those are used for the next generation. So that way we have a good chance of that next generation having the genes that would contribute to their extreme longevity. So
they have to at least survive beyond a certain point to be considered
exactly
the next
exactly.
Cool.
Exactly. Yeah.
All right, cool. Well, thank you so much for coming on the show. We want to we'll be checking in periodically as we learn new things and things like that. Grace, thank you so much for coming on and all the great questions that you had today.
Yes. Just kidding. Thanks, everyone.
Very funny, Grace. All right, everybody. Thanks a lot. We'll see you soon. Remember to check out episode 4 with Cory Doctorow that also went live today. And we'll see everybody later, and sweet dreams. Bye.
Bye.